Hi, everybody. Good evening. Just confirming again if you can hear me all right. And welcome to the second live session for the course Cell Culture Technologies. I'm Pallavi Gupta. I'm a PhD student at IIT Delhi, and I'm a TA for this course. So we met last week at the same time, Monday, and we talked about what was covered in the first week. And today, this is like a live problem solving session for each week, where the main idea is that we'll be going through some of we'll try to cover everything that was covered in the course in the recorded lectures. We'll have a quick summary of all of that. And if you have any questions, you can ask that. This is meant to be an interactive session. So please feel free to unmute yourselves at any point of time. And if that's not possible, then please definitely use the chat feature. Um, Type out your questions, anything, absolutely at all. Um, and then so once we've discussed the topics from the week's lectures, and then once we have clarified the doubts, we'll go on to solving some practice problems, which would be similar to what you have in your assignment for that week. So we'll have some 5-7 questions um, that give you an idea of how to solve your assignment questions, because the deadline for these assignments is every Wednesday. So a Monday class, it just helps. And then we will also talk about the previous week's assignment because the solutions have been released. So we'll talk about that, and if there's any confusion there. And again, please feel free to ask any questions. Before we start today's session, in case there was, so the last time we started off by introducing ourselves. Some of you told me what you're doing right now. So that would just help me in getting an idea of what your background is. So what sort of details would you be looking for, or what things are already clear, and then we can build on those. Because in these sessions, we don't need to necessarily restrict ourselves to what was covered in the lectures. We can cover a few things here and there also, depending on your interest and if I know about it. All right, so before we start, would any of you like to tell me about what they're doing currently and why they took up the cell culture course, or what they are, you know, what do they expect from this course? Also, if any of you, I can see some familiar names from last time. So if any of you have any questions or suggestions for the first set of live sessions, let me know. We can spend a few minutes here and then we can go on to discussing about this week's topics. Anybody? I'll wait a couple of minutes. In the meantime, I'll start sharing the screen. So again, the more interactive this is, the better it'll be for everybody's understanding. But that's okay. It's totally up to you. No problem. All right. You should be able to see my screen now. Okay, so if there is nothing to talk about right now, then I'll just go ahead. So just some housekeeping information just so that everybody's on board. This one is an 8-week course, so there'll be, and there are a total of 40 lectures, roughly half an hour each. So that's 2.5 hours worth of recordings that you get every Friday. So the first week's done. The lecture videos were released about like some time back. We had a couple of live problem solving sessions. So I do the ones on Mondays, and if I'm right, there's another one on Tuesdays, which is taken by the other TA for this course. So you are free to attend either of these. You don't necessarily need to attend both. We would be covering similar topics. And of course, the attendance in these live sessions is not going to impact your overall performance of the course. For the NPTEL courses, especially for those of you who are interested in getting a certificate, it's your marks in the assignment and your final exam that would be important. These sessions are just to aid your understanding. So we're done with the first week of lectures, which I do on Zoom, the other ones on Google Meet. All of these sessions will be recorded, and the recordings will be uploaded on YouTube, and you have the links, you have the access to these videos. So if you go to your course page and you scroll down, there's a tab on the left which says problem solving sessions. You go there, it redirects you to an Excel sheet where you have the lecture video recording links, and you have the Google Drive link where we are going to upload all of these PPTs or whatever material is being discussed in the class. And of course, there is a question Q&A forum also available. Make use of that if any of your questions remain unanswered in these live sessions. All right, so we have 17 people who've joined us. That's nice. Again, attendance is not important. So in case you're not able to attend the entire duration of this session, that's all good, no problem. Okay, thank you, Samriddhi. You're a biotech student. Great, good. So if I may, again, feel free to say no about it, but would you like to tell me if you've had any practical experience of cell culture so far, or would you have some in your in the rest of the course, in the rest of your program? Do you know that? Let me know. Yeah, and because uh, from this course, of course, in this online format, we're not doing any hands-on work, but this is to give you an idea of what cell culture looks like, what things should you keep in mind, how did this field develop, what all do we know, what all do we not know, and yeah, okay. And others along the duration, like throughout the session, if you feel like you would like to also tell what you're doing currently, that'll be great. All right, I have the chat window accessible. Okay, great, great, great. Yes. So when you're doing, again, the first few sessions, like if you also saw last week, we're mostly talking. There's not a lot of, I mean, of course, there's practically no numericals that we're doing here, or there's no derivations. It's not a math um, course or a statistics course. So we'll be talking more about what it is and how it is. So yes, you will get an idea of what cell culture is, how do you deal with this. So I remember, so I started doing cell culture about 2.5 years ago. And I remember before I got into this field, somebody had told me that, oh, cell culture is terribly difficult and you have to be very, very careful, and there's going to be contamination and you have to keep yourself alive. And this is what I had heard. I got scared that, okay, maybe it's very tough, but okay, let's see how it goes. And then when I did cell culture, of course, it's tough. You have to be careful. You can't be careless. But then it's not like you're going to be killing yourselves every day. Mistakes happen, but as long as you're careful, and for most cases, there's a protocol. There are well-established protocols. So especially when you're starting, you have that. So you're working with known things, and then later on, once you have had a few months or a year of experience, and then you can play around with things if you want to develop some different protocol, if you're looking to do some customizations. We will briefly touch upon this, and I would just like to tell you. So, thanks for asking this question. So, my PhD work is on 3D cultures. I am growing something called mini brains or organoids, brain organoids. So, if time permits, along the way, along in whichever classes, wherever it's relevant, I can tell you a few things about it. Again, I'm no expert at that. I'm also a student. I'm also learning and currently doing that. But we can talk about this then. So yeah, thanks, Kashish. Thanks for the question. So we'll talk a little bit about um, so 2D cultures for sure. 3D, we'll go and see where all it can be fit. All right. Okay. Okay. So this we'll just take a quick recap of what was covered last. Oops, sorry. What was covered last week. So what cells need to grow is oxygen and carbon dioxide supply, and so intake and um, removal. They need a substrate to grow on. Most cells that you talk about in cell culture, they need a substrate to grow on, and they need, a lot of other things where we talk about nutrients, which would be so. Let's just say you're dealing with two types of cells, okay? Heart cells and brain cells. They are cells after all. They're all eukaryotic cells, human cells. So their basic requirement would be similar, but there is something which is making them heart cells, and that is making them stay as heart cells. Okay, so like you say that different people of different ages have different requirements, or people from different places around the world, they have different preferences. Just like that, the basic idea is that yes, they need food, some sort of food, but what exactly they need, what is keeping them in that happy stable condition of what they are, that would be different. Sure, Kashish, of course, sure, I'll share that with you at the end of this course, today's session. Yeah, cool. Oops, sorry. All right, so yeah, so the okay, so but when you talk about, so we came across these two words, in vivo and in vitro, or in vivo and in vitro, whatever you prefer to pronounce it. So normally there's a dynamic environment that is provided by their neighbors. And when we talk about neighbors, they could be the same cell type. There could be one other cell type. There could be two other cell types. There could be multiple other cell types. And or the cell types can be stationary or they can be moving. For example, if one of the neighbors that we're talking about is blood, well, it's more dynamic, right? Or indirectly, it's blood that is controlling the dynamic nature of any system in an actual organism. So, of course, there's a dynamic system. We don't have that yet in cell culture. Although, just to put it out there, we people are talking about flowing blood through a lot of these cultures, and to make it more similar to what is there in the real world. But again, it comes with its own problems, because the more dynamic, so the more complexity you do. So for example, that's where we come to the second point here. So when you're talking about one pure culture, let's say cell type number A, it needs a particular set of things. Let's say you know what exactly those things are and you're able to provide it. Okay, perfect, fine. Second case, if you have a combination system, maybe you're growing two or three things together. The good part can be that maybe one is going to provide something already, so you don't have to externally provide it. Okay. So cell type B releases one of the things which you would have ordinarily given if you were just growing cell type A. So externally you would have given it to cell type A, but now that A and B are together, B is already providing them. So that helps. Okay. But then of course the complexity comes that B needs something else. You need to provide that. And then there could be certain things. So, and then not every cell type might want to grow with the other cell type. Okay, they could have very different nutritional requirements. They could have different cell dividing rates. So let's say you start off with 10, whatever, 10 cells of one type and 100 cells of the other type, but the one that is 10 can take over, right? So then does it help you to grow both of them together? Things like that. So there could be a lot of things. Again, these are just, I'm just, throwing words at you at this moment, but just so that you keep this in mind. Versus, and ideally, what would it be to have a self-sustaining system? That yes, you have a blood supply, and you have cells, and then it's the blood is taking up the waste. Maybe you also have something like a filtration system that's able to remove that waste. So you're making now, so right now, like what we were talking about in the chat, we mentioned something called organoids. Again, this is not a part of this course, but when you're talking about growing one type of cell versus two types of cells together versus three types of cells together versus when you're growing maybe 10 types of cells together. So you are becoming, you're going closer and closer to what an organ would look like. So rather than calling that thing an organ, we call it an organoid because it's similar to an organ. Now, but one organ cannot exist independently just by itself. It needs something else, and that maybe needs something else. So all of those together. So people use this word called assembloids, where you're assembling multiple things together. Ideally, we are moving towards a place where we are making a self-sustaining system with multiple organs, multiple cell types. But yes, let's see how it goes over the years. So depending on what sort of a system you're looking at, the media that you provide, the growth factors you need to give. Growth factors are small molecules that you need to give, or hormones. We'll talk about these in more detail in, I think, week 4, if I'm not wrong. But yeah, we'll talk about it then in detail. Um, so the basic requirement that all cells have is they need energy and they need carbon source. So cells are making a lot of things, right? They're making their own DNA, RNA, proteins, and a lot of it is all carbon based. So they need carbon, and that carbon source is glucose. Okay, C6H12O6. That's the formula for glucose. So that definitely is there, going to be there in all the media. And what this image here is showing you is that this is an example of a culture plate. And if you can count here, so there are 6 wells, okay, 6 depressions in this plate, 6 by 4 depression in this plate. So that's a 24 well plate. That is a lid for this plate here. And this is a bottle, this is a reagent bottle or a media bottle, which has this media. And you're pipetting the media into each well, wherever the cells are. And that's just a reference image. In terms of the pH, so this will be dealt with in more detail when we talk about the media. But you have to be careful that cells in our body, everything is happy when the pH is around 7. It's a little more than 7. Again, we'll talk about the details of why and how when we come to it. But still, the pH is something around 7. And that is what all the cell types are okay with. Some cell types can tolerate a different pH. For example, the stomach cells, which are releasing acids, they can handle an acidic pH. So acidic pH is less than 7, basic pH is more than 7, pH scale is up to 14. So, but also these cells, they cannot tolerate that acidic pH for too long. So just something to talk about here is you're seeing that this media, and also when we talk about later, media would generally look like this sort of a color. It's going to be something like a red color. But normally media is a lot of water and sugar and salt, right? So where is this red coming from? There's no red component that you're adding as such. If you look at the constituents, so this is because there is going to be a pH indicator in this. So right now, so I think this is phenol red. So basic to neutral, it's going to be pink. And then as and when it gets more acidic, which it will because it's in a CO2 environment. So CO2 gets incorporated, you get more and more carbonic acid being incorporated here. This turns more and more orange and yellow. Okay, so a pinkish reddish color indicates that, okay, the media is fresh, the cells are happy, the cells are there. But then as soon as the media starts turning orange or yellow, then you know it's time to change the media. Changing media generally means that here you have this plate, right? Now, if you remember from the last time we talked that at the base of these plates, you are adding some sort of an ECM, extracellular matrix. Okay, it's almost like an invisible layer. In fact, some of these culture plates, they come with that invisible layer already there, right? This is a plastic plate, so you can keep it in the shelf. But if there was a coating on this, it would be shipped, it would be transported at a different temperature. Anyway, that base, so it's an almost invisible, a very thin layer. The cells are going to be attached there. Okay. So when you're giving the media, the cells are actually, they're not floating in this media anymore. They're still stuck, but there's a generally, let's say that we're dealing with a single layer, a mono layer of cells. So this is where your base or that ECM is. This is where your cells are sitting, and on top of that is where your media is. Now, when you want to remove the media, you can just pipe it out the media from the side so that you're not scratching the cells. You take out the media and immediately you put a new media, fresh media back. So the cells are without media for a very little time. What people also do is that you can leave behind a little bit media there so that the cells are never dry. Because of course you're dealing with multiple wells at a time. So you're going to remove one at a time from 24 wells. However fast it is, it will take you half a minute, okay? At least it's going to take you 1 second per well, right? So until you come back to the first well and give it fresh meat, it's going to be without anything. So it's going to be dry, it's going to be without food for 30 seconds. So let's say if in each of these wells, the amount of liquid that you see is half an ml, you can maybe just, on an estimate, you take out three-fourth of it. And then you can put in fresh media about half a mil. So at any point of time, it doesn't have half ml. Maybe it has 600 or 700 ml, microliters. Sorry. Yeah. So everything that I was talking about, half ml or 600, 700 or 500 microliters. Okay. Um, that's about media. So yeah. So that's where you would need to change the media when it starts turning yellow. And of course, it's just a comment from the last week that you are growing cells, and if you're using whether or not you're using a standard protocol for a particular type of cell, it's always important to check if your cells are behaving the way they should. There has to be some sort of a readout. If you can just look at them under a microscope and see, then that's great. Okay. If you expect the cells to be circular and they are circular, well, there's a good chance that everything is fine. Okay. But so what generally people do is when you're working with some kind of cell for the first time in the lab, that is when you need to do all of these characterizations and checks. Now, once you have established, okay, this kind of cell, this kind of protocol in our lab is working the way they published in a particular paper, then you're good. Everybody else in your lab doesn't need to necessarily do this. All right. Okay, one quick question that I have for you, if anybody would like to answer. So, like I said, this is a plastic plate. Um, and so there is this lid that you see here, okay? So if you're using a microscope to look at the cells, tell me what sort of a microscope should it be? That is, so there's something called an objective in a microscope, which is the main lens through which you're looking at your sample. Would it help if that objective is above the plate? That is, it is towards the lid of the plate, towards the cover of the plate, or if it is at the bottom, if you're viewing from below, or if you're viewing from the top. Does anybody have any idea on this? What do you think should it be? What sort of a microscope works? Whether objective is at the top or the objective is at the bottom. Inverted microscope below the plate. That's perfect. So the idea is that when you're looking from, so, okay. Now this, right now what you see here, the plate is open and you're not worried about contamination, as we'll just talk about in a few minutes, because right now you're working in some sort of a safety cabinet, some sort of a hood. We'll just call it a hood for now. It's a safe place where your cells are not going to get infected. So right now I'm in my hostel room, okay? If I have a culture plate here, and if I'm working with cells, the very next day there's going to be something growing on it. Because this media, this is food. This is very, very rich source of food, nutrients. Everything, if you have some spores, something floating around here in the air, everything would like to go and sit there and grow. But of course, when you're changing the media, when you're adding media, working with cells, you have to have the lid open. When it's there in an incubator, as we'll again talk about more about it in a few minutes, there it's always closed. So whenever you're taking this out to a microscope, generally microscopes are always outside of these safety hoods, whenever inside the hood. You can have microscopes inside the hood too, but that's rarer. So a microscope is outside, so it's not in the safe place, but you have to image your cells. You have to take a look at your cells. So if your objective, if that lens is on top, you will not be able to focus on these cells because the cells are sitting here and your objective can maximum go until wherever the lid is. So there's still like this 1 centimeter to 2 centimeter of a width that you're away. So you will never be able to focus on that. But on the other hand, if your objective is below the plate, so if, for example, if this is the plate that you're working with, my objective is going to be here. So at the bottom, And well, you don't have to look through it through the bottom. It's all connected. So your eyepiece would still be at the top, but you're getting the image from the bottom of the plate. So what you're looking at is actually inverted, but it doesn't matter because in most cases you are looking at a single layer, or even if you're looking at multiple layers, you're still looking at the layer that is the first from the bottom. So this difference it's not a problem. But yes, so just keep in mind, more often than not, you will have an inverted microscope in a cell culture setup. Okay. All right. So yeah, so here all I wanted to say this was that keep in mind that you have to characterize your cells. You have to keep in mind that the cells should behave what you expect them to behave. That is when you're using a known protocol. When you're making a new type of protocol, that, okay, let's say the current protocol, it takes 30 days to grow a particular type of cell. You're like, I want to grow this in 3 weeks or 2 weeks. I want to half the time. So you're going to do something else, maybe add more growth factors, add something that fastens that differentiation. Let's say if you're differentiating cells, whatever it is, you're doing something different. And maybe what you're getting is what it looks like that particular cell type. But in the process of adding more growth factors, you don't want to mess up something else. So a lot of these characterizations then are needed to be done. All right, we'll come to more of these things again. So a large part of what was covered this week, sorry. A large part of what was covered this week is about the design of a cell culture facility. So in general, yes, this is all cell culture, but why would you do a cell culture? Why would you set up a cell culture facility? Why is there a cell culture facility? Could be for different reasons. The first thing is what you're doing there. So more often, what you see in a university or in a university sort of a setup, in an academic setup, is maybe the people are interested in studying the biology. There could be developmental biology of the cells, okay, or of cells of organisms. Similar to that, it could be that you're studying the effect of some infectious agents, viruses, bacteria, okay. You may be infecting human cells with some bacteria, you may be infecting human cells with some virus, okay. Or it may not be a human cell culture facility, it could be culturing some other cells, right? It could be a non-human animal, it couldn't be a non-animal sort of a cell culture facility. We won't be going there, we'll be restricting ourselves to mostly humans and a little bit more in general about animal cells, but still, it is not going to be restricted to just humans, okay. Mouse, rat, monkeys, a lot of that is also done. Then, whether you are making something known as transgenics or genetically modified organisms. So, if you are making a change in the DNA of a cell, then that classifies as transgenics or genetically modified organism or GMOs. Okay, that is all that there is. You might have heard about CRISPR. Okay, the good, the bad, the everything about CRISPR. Again, let me know if you would like to talk about it and if time permits towards the end of the session. But yeah, so that is what is done. Because if you want to study the effect of a gene, okay, when we say you want development, you're studying developmental biology, you have a hypothesis that, okay, probably this gene is what is doing something. Okay, then the best way to find out if that's actually the case is knock out that gene, remove that gene, and see if what you thought was happening was actually happening, right? So, for example, if you think that, okay, this you don't appreciate, let's say, the task, the work that the cleaning staff of a particular place does. One day they take a leave and you'll see that, okay, everything is a mess. There's rubbish everywhere, it's not clean, there's dust and everything. So you realize that yes, they were indeed doing their task. So similar to that, in a healthy environment, you remove a gene and you see that, okay, now a certain function has gone, aberrant, it's gone haywire. So it's most likely because of that gene. Okay. You may be studying when you may be studying a particular disease. It doesn't have to be infectious, but it could be just a disease that you're studying. You may be doing some tissue engineering where it's this is more medical related, but you're growing tissue on a substrate. You may be making some sort of a biosensor. We briefly talked about it last week also. Uh, okay, and then these two sort of labs, these are generally more industrial scale. So these media and these gels or the matrices that you use for cells, more often than not, this is what people buy directly. So you buy media. These are, you know, yeah, you get bottles which are like 500 ml, half a liter bottles, big bottles like this that you can order. Then just keep it at 4 degrees or minus 20 degrees, and then they have an expiry of maybe a few months to a year. So you just directly use that. So people are making that, they're optimizing it. You do get like newer versions that this is supporting better growth. It just gets better. Okay. So medium or substrate is there. There could be you could have labs which are screening some materials. So for example, we talked about this last time also, which is and calling it something like personalized medicine. It can be more than that also, but just giving you an example of that. So you have cells of a particular type and you're testing some drugs on it. You're testing some medicines on it. That, okay, is this killing the cell? Is this not killing the cell? And so on. So that's some sort of a screening method. Whether you are using animal cells or not. So where are you getting your cells to work with? What cells are you working with? If they are human cells, again, human cells are these primary cells. Have you just got them fresh from some hospital that did a biopsy? Or were these frozen? Or were these cell lines? Again, we'll talk about more of these in detail. But what sort of cells are they? Or are they coming from an animal? Are you dissecting that animal then and there? Again, we'll just cover a little bit of that in today's class. So that is about the design of, like, that's the objective. And then that this entire thing leads to what sort of a level of sophistication are you looking at? So, of course, if you're working with infectious diseases, you have to be more careful. Something basic is not going to work there, okay, because you have to be careful that whatever that infectious agent is, it should not escape. Okay. So I don't know if any of you remember, but when during the initial days of the COVID-19 pandemic, there was a speculation, which I'm still not sure if it got clarified, that this emerged in one of the labs in Wuhan, right? So whether or not that is the case, that so the thing is that the first thing is people are working at a particular place. So of course, there's always, you can never say that there is absolutely zero chance that an infectious agent escapes. Okay. But you have to minimize that as much as possible. A lab here that does cell culture, which is just working with cells and not an infectious agent, you can have a little bit more leniency there, right? So, and of course, by the level of sophistication, we mean what sort of equipments are you talking about? Um, Big, how what scale are you talking about? What equipments you need? How many of those equipments you need? So again, all of this is very related. So do you have some plan for expansion? So generally, again, the example that was given in the recorded lectures, that was let's say you're starting off as an assistant professor today in a lab at some institute. And today you think, okay, I will probably take 2 or 3 PhD students and maybe a couple of master students and maybe 1 or 2 interns for the first 2 years. That's all what your lab is going to look like. But 20 years later, maybe you have 2 postdocs and you have 7 PhD students and 3 or 4 master students, and you keep having interns and project students with you. So that's like a capacity of around 15 to 20. You have that sort of a space. And there is always a space crunch wherever you are, whether it's India or whether it's outside. Once you've got a space, it's very hard to expand that space unless you are an independent body, right? If you're in the lab, it becomes very tough. So you have to maintain, can you show that you're occupying this space, but you actually just, you currently just require this, but use this much so that whenever there is an option or whenever there's a need to increase, you can actually increase. So yeah, space constraints, space needs to be managed, financial constraints are always there. So what helps in all of this is whether the institute or wherever you are, does it have some centralized facilities or some central facilities? For example, in your lab, you can maybe have two normal microscopes, one of these inverted microscopes that we just talked about, and maybe another one can be like a fluorescent microscopes that you, because there's a good chance in all of these things, you'll work with some fluorescent molecules to label your cells. Again, we'll see when this comes in the course, and we'll talk about that more. But simple thing, you would have seen images which look like these shiny cells, red color, green color, blue colors. So more often than not, this is something you would need, just for 2 minutes in a day. You just quickly want to take a look what your cells look like. So you don't want to go maybe like 5 kilometers away just for it. And when you're working with cells, you also cannot do that. Your cells, they need a particular environment and they need to be sterile. So you cannot go to a facility that's 5 kilometers away, right? You may have a facility which is maybe like in the next building, some sort of a central facility where maybe you're not working with live cells there, but you can put these cells, kill them, which is called fixing cells. You can fix them on a small slide, these glass slides that you have, and then image them somewhere else. So that sort of a thing can still be done with cells, okay, when they're no longer living. So that helps. Then all those fancy big microscopes you don't need to have in your space. So that saves space, that saves a ton of money. Okay. So of course, how many users are there? Um, Again, what do you envision the capacity now, later? And you have to have space per person. You cannot have 10 people when as per rule, you should only have 4 people in that space. So typically, what sort of a space we're looking at is a 4 by 2, 4 feet by 2 feet space. So 4 feet like this. So I have 4 feet working space for me because, okay, I will have 2 or 3 plates with me, these culture plates. I'll have some pipettes. I'll have my media bottles here and there. So that's 4 feet. And then, of course, 2 feet is this width. So that's like bench space. Okay. So overall, again, all this is all very qualitative information just for your reference. And this is now we come to what precautions do you need. What do you have to be careful with? So you don't want things to be congested. You don't want people to be running into each other. Okay. So let's say you have a bench here and a bench here, but then you have to remember that 2 people are going to be sitting here and working on that bench, right? So you don't want space that there's no working space for them. So this bench cannot be like this. The 2 benches need to be at least this much away. So something like that. So, and of course, bench height and all, it goes. So again, a lot of this is more important if you yourself are setting up a system, which is a little far. So of course, when some of you, no, when all of you, whoever's interested, when you go to places, see 1 or 2 or more culture facilities, and then you can compare that, okay, this was good there and this wasn't great here, and this could be improved. How could this be arranged? Things like that. Okay. Safety paramount. So while a lot of focus goes into when you, like as we'll discuss today and also in the upcoming classes, a lot of focus goes into keeping your sample safe. Okay, you don't want any contamination. We'll talk about different types of contaminations, but it is equally important to keep yourself safe. Okay, because there are hazards in a lab. Very often in these big labs, you will have a proper induction that, okay, you will have access to those labs only after you are taught that, okay, this is where you enter from, this is how you exit. You have to wear a lab coat. You have to wear goggles, safety glasses. You have to have your gloves on. You have to keep washing, like wash your hands before and after. Keep your countertop sterile. Use ethanol to spray. Change gloves if you think. Change pipette tips. All of this. Uh, so a lot of this, like halfway, it goes into keeping your material safe, but then a lot of it goes into what you need to do to keep yourself and everybody else safe. Okay, so storage shells, again, you work with these culture plates, right? Today you're using a culture plate. Now once your cells have used up that entire space in that well, you have to take those cells, take a part of those cells and put them in a new place so that they have more space to grow, right? You can't just remove some cells and allow the other ones to keep growing like that. That's not how cells will stay happy. You need to transfer it at a new place. So when you see this, so let's say there are about what, maybe 14 of these wells that are full. You have to put all of them into a new plate now. So let's say on an average, people would transfer these things every week. Okay. So every week one person needs one plate at least, right? But of course there'll be more people. Some people would be working with 10 plates at a time, some people would be working with 5 plates, and so on. So you need a lot of these plates. You can't be ordering one at a time. Okay, so you'll order them in like big batches, maybe 50 plates. They come all together, there'll be big boxes. Where do you store those, right? So you definitely are going to have storage shells. Very often they'll be below, but very often they'll also be somewhere at the top. So when you're taking out the shelves or taking out these boxes from these shelves, you have to be careful. Okay. We'll talk about cryo storage in detail in a few minutes. I have a specific, like a dedicated slide for that. There are going to be a lot of heavy equipments that you'll be working with. So let's talk about these two here. Laminar flow hood, again, we'll talk about them a little later today. So cells, they need a particular temperature, which is 37 degrees, which is understandable, right? Your cells in the body need 37 degrees. So the cells you're growing in the lab, they also need 37 degrees. Now, they need some amount of CO2. Now, I will not explain in detail, but that CO2 is important to keep the pH at around 7. So the media that you give these cells, they are buffered, so they can resist some sort of changes in the pH. And to allow all of that, you need CO2. Generally, it's around a 5% CO2 that's given in these incubators. So you have incubators connected to a CO2 supply, carbon dioxide supply. Now, that supply can be either in-house if you are the only lab that's doing cell culture. So you'll have a one or two CO2 cylinders with you, and there's going to be a pipe that's traveling from the cylinder to your incubator. These incubators, which are at 37 degrees, and they'll have some option to um, manage that. They'll have the option to control that. Okay, this is 5%. CO2 is only there. It'll keep it between 5, 5.1, 4.9, like that. So, but also in some parts, suppose the entire building is dedicated to doing cell culture, then it doesn't make sense that every lab has their own CO2 supply and you keep filling and refilling CO2 cylinders every week or every 2 weeks or whatever the time it takes, right? So instead, you can have a centralized CO2 facility. So that's being managed by, let's say, that department or that building. And then you just need to ensure that, your pipes and all are connected fine. All right. I've been speaking for some time now. So if anybody has any questions, any confusion, suggestions, clarifications, let me know. Also, if I am wrong at any point of time, please correct me. Cell culture, like I told you, I've been doing it for about 2 years now, and I'm doing just a very small part of what cell culture, is composed of. So it can happen sometime that I may be wrong. So if any of you catch that, please feel free to let me know that I've said something wrong. Okay. So if there's nothing else, then I'll just continue. Now, airflow is an important part in a cell culture laboratory. So generally, we talk about so if possible, then your cell culture lab should be at a negative pressure so that there's nothing. Negative pressure means it would be at a relatively um, lower pressure so that it's going to be low. Yeah. So it's going to be so things from high pressure to low pressure, right? So yeah, so it's going to be at a low or a negative pressure so that nothing goes out from that lab. You want, so air can come in, and air generally you can have filters, some sort of a filtered air supply that's coming in, if possible, not from the door, but from otherwise from the air conditioners and everything. But generally, you would want the air to be not escaping from that. I use stem cells for my culture. Yes, I use embryonic stem cells. Yes. Yeah. So the airflow you want, so the airflow should not go out, okay, from the system. So that's one thing. Just keep this in mind. The other thing is there's going to be one or more doors that lead to where you're working, right? So the incubators and your laminar flow hoods, these hoods where you're working, these things should be away from the main door so that there's no air coming directly. Suppose I am working and I've just opened the incubator and I'm putting my cells in, and then somebody has just opened the main door. So that main door air, which is unfiltered right now, it's just coming in and it's there. So although my cells are all, they're all closed and everything, but you can never be too careful. Okay. Yeah. All right. So then you want to talk about when we say incubators. So, okay, while every cell, let's say all the cells that everybody in a particular lab needs 37 degrees Celsius and 5 degrees, 5% CO2 for their cells to grow. But would you want to grow, let's say I am working with I'm working with human cells, just pure human cells. I'm not doing any infection on them. And then somebody else in my lab, my, let's say my colleague, they are working on some sort of a bacterial infection on human cells. They also want 37 degrees CO2. I also want 37 degrees CO2. But since theirs is a bacterial infection culture, I don't want to grow it in the same place. Okay, because what if something goes out from their incubator, from their culture plate, and it infects mine, right? So what if I've been working for 2 months and it all goes? So then, while let's say there's not a lot of people, so I'm never fully using up the incubator. So when I say incubator, it typically has, so it's, imagine it to be like a fridge shaped thing. You can have multiple shelves in it. That's all. Okay. So you can maybe put, let's say you can put 20 plates in that. Okay, it's a small incubator, you can put 20 plates inside it. And I or whoever is using it in the lab, at the most we have 7 to 8 plates there. So you're not fully using up the incubator. If the people who are working with the bacterial cultures, bacterial infection in human cells, if they can also put it there, and we would just, it would be sufficient for us to keep one incubator. But well, I don't want to risk contamination. So it's better to invest in two incubators rather than investing in one and then ending up wasting your culture, which itself is going to be expensive, right? Because you've spent time, you've spent a lot of resources on that. So you don't want that. And sort of the same thing can go for incubators also, for these laminar hoods. Because what if somebody did not clean it? I'm using the same space essentially, right? Somebody has just worked on bacteria, and then now I am coming, I'm using the same pipettes and everything, and what if I infect my culture? So typically, not just incubators, but also these laminar flow hoods, which about which we'll just talk in a few minutes, would also be different. Oh, yes, that's a very good question. So incubators do have water there because you don't want the cells to be very dry. So two things. There would be this sort of a humidity. I'm not sure. I think that's also 5%. sorry, 95% humidity or something is what is recommended. So briefly, what I'll just want to explain you is, so if you, so okay, there's a difference between, so are you, so you're familiar with what evaporation is, right? Even when the system is not at a very hot temperature, water will still, it gets evaporated and it still gets converted into vapor. Now, when you're talking about an incubator, there's the 37 degree, which is quite high, right? 37 degrees when in summer months it's 37, it's hot. Okay. So you don't want that if there's no water, the cell medium, the medium in your plates, that is going to start getting evaporated and it's going to condense in your culture plates. And also the culture plates are not perfectly, they're not sealed. So your water is going to escape. So your media is going to get depleted soon. So you don't want that to happen. So there's going to be a little water. Again, that has to be sterile water. Yeah, 95%. Thank you for confirming. So you don't want that to happen, right? So that's why you add water there so that it's a good amount of humidity there. Copper sulfate. I am not 100% sure. I've never heard about copper sulfate, but if I like what I have used is just like a disinfecting agent, but I think copper sulfate would do the same thing. I think it has to be something to do with disinfection. But I can confirm with you. I'll confirm this for you. In the meantime, if anybody else knows what the reason is, then please just yeah, just everybody please anybody who knows, you can just put the reason in the chat box. I'll just write that. So copper sulfate and water. If time permits, we'll just quickly Google this at the end of the course, end of the session. Okay. So that's there. Entry and exit should be very, very clear. You shouldn't keep boxes at the entry exit because just in case of a hazard, you want to exit quick. Okay. Fungicide, that's possible. Yeah, it has to do something with some sort of making it sterilized, like sterilizing it or disinfecting it. But we look at it. Yeah. Waste disposal or trash disposal has to be very appropriately done. So again, there are these ideal objectives, and then it depends on how much can be achieved. But a lab would generate different types of waste. So one waste is going to be, so when you're doing cell culture, you would also be doing non-cell culture work. For example, once you have extracted some DNA or RNA or protein from your cells, you're no longer working with cells, right? Now what you're doing is just basic molecular biology, right? So in that case, you don't have to be, you're not working with anything infectious, you're not working with food media. So you don't have to be, you have to be careful, but then you can, again, the level of carefulness can decrease a little bit. So the waste you're generating from when you have live cells with you versus the waste you're generating when you're dealing with just molecular things, that's different. And also you can have things which are just plastic waste. So your cell culture plates, your pipette tips, right? Tissues, gloves, these would be all plastics. So you don't have to be, again, they are not sharps versus you might generate a waste that is sharp. So when you talk about pipettes, broadly we'll categorize them into two types of pipettes. One is a pipette which has these up to 1 ml. These are called micropipettes. So 220 microliters, 200 microliters, which you could just, the common ones that you see. And then there are also these bigger pipettes, which can, if you want to meet, especially when you're working with media, you want to work with 10 ml at a time or 50 ml at a time. There will be bigger pipettes, which are called pasture pipettes. So which are glass. So these are glass pipette tips. So if you are using that, then glass, that's of course a sharp, or you may be working with blades. Okay, so all of those things. So in different places, you may want to segregate them separately, because glass and all of these, they can tear through whatever waste disposal bag, trash bag that you're talking about. That can be different, right? You you can't, you shouldn't ideally put the things together. The sharp things should ideally go into two bags. So you have a bag, you put that bag in another bag, so it's safer. Okay. Lab coats, and say, I mean, of course, lab coats, gloves, um, safety glasses. So gloves for sure. And a lot of things, again, it depends on resource management, but whether or not you can reuse gloves, ideally you should never. But okay, we'll take that with a pinch of salt. Uh, okay, do we have something? Yeah, it has to be some sort of bacterial or fungal, some growth. Yeah. Okay. All right. Next up, again, a lot of this is just repeat, but I've tried to summarize whatever was discussed. So there were, if I'm correct, there were three lectures where all of these things were discussed in more detail this week. So it's just a summary of everything. Laminar flow hood and incubators. So laminar flow hood we'll talk about. We'll talk about in detail about the liquid nitrogen cryo storage. So cryo, the word is about cold, super cold things. So we have like two dedicated slides for that. CO2 incubators and cylinders. So yeah, so the idea of a cylinder is these are heavy things and they're not very stable to be placed. Okay, it's a big cylinder, maybe up to like an average human height, and they're going to be heavy. So you might want to roll, like people generally roll it in and out. Best cases, you use a trolley for this. If there's a centralized facility, things get a lot more simpler. But yeah, so if there are cylinders, not just like you don't want to just keep them against a wall, but you'll want to patch it, like clamp it there so that it doesn't fall, right? Even if the balance is a little here and there. So liquid nitrogen generally always required when you're talking about cells, but more commonly you'll be working with these temperatures. So 4 degrees. So 4 degrees is what you normally have in your home fridge, okay, where you're keeping your food cold but not frozen. That's generally 4 degrees, something around 4 degrees. In the labs, -20 and -80 are sort of like the set temperatures. Uh, of course, there's always like in most cases you have a control option that okay, you would like to keep it. You can keep it to -70 or also, but generally -20 is there in freezers. So also in the freezer that you have in your home fridge, it's generally -10 or lower. So -20 is what you see in the labs, and -80 again is another thing that you see in the lab. Generally, -80 freezers are -80 all throughout. Okay. Yeah. Now, why would you need all of these temperatures? So again, two main kinds of thing, or let's say three main kinds of things that you would keep in a lab. One is, of course, your cells. Okay. The second thing you would keep is the media and media related things, whatever you are adding to grow them, whether that's your the ECM that you're adding or the growth factors and all of that. And the third thing is whatever you are extracting from those cells. Okay, you might be taking out DNA, RNA, protein, something from those cells, right? Where do you want to keep those? So 37 degrees is where the cells are growing happy and healthy, but that is also the temperature where you'll have other things growing. So that's why you have to keep them very safe. And that's also the temperature where things can get, where a lot of these enzymes and a lot of words which end in A's, so RNAs, DNAs, protease. So a lot of these enzymes, they also work. And these A's enzymes, these can chop up the respective things. So DNAs cuts up DNA. Okay, it's going to kill, it's going to just, mess up what DNA you have. So it's going to spoil it. RNase is same, protease is same thing. So the lower temperature you keep these, the more safe they are. So because these extra enzymes, they cannot work there. Okay, so 4 degrees is okay for a few things when you're keeping them at a for some small time. Like these media bottles, they're good at 4 degrees for a few weeks. A lot of these things that you would need probably once in a month, you might want to keep them at minus 20 degrees. And um, yeah, so your DNA can be kept at minus 20. DNA is okay at 4 degrees also for some time. RNA, you would definitely prefer to keep them at minus 80 because they're really, really susceptible to being spoiled. Yes, so trypsin and FBS we can store at minus 20. So a lot of these things. So trypsin is so it's an enzyme that, correct me if I'm wrong, but that is what you're using to detach your cells from that ECM where they're stuck. Okay. And FBS is a fetal bovine serum. So basically that's a serum that's coming from fetal cows. Okay. That is one of the important things that you want to provide cells for them to grow, right? Yeah, okay. So yes, so everything, so all of these things, they have their particular temperatures. It's there in the bottle, and that is where, so it says that, okay, at 4 degrees, temporary storage. So what people normally do is, there's a word called aliquots. So let's say the bottle that you ordered, it has 10 ml of something. Okay, but let's say today I'm just going to use 1 ml out of it. And then next week I'm going to use another 1 ml. And it says that you should keep it at minus 20 degrees, right? So now, okay, today is at minus 20. Now minus 20 is going to be frozen. Okay. I cannot take out 1 ml out of that 10 ml of frozen block. I need to thaw it. So thawing means I'm going to make it liquid. Generally, that's done at 4 degrees. So I'm taking a thermocol box. I fill it with ice. I put that bottle in that. Maybe it's going to take me 30 minutes to thaw it. I mean, I'm not doing anything. It's getting thawed on its own. Then I take 1 ml out of it, use it. What do I do with the rest 9 ml? Would anybody like to say what would you want to do? So I have 10 ml. It's said that you should keep it at minus 20 degrees. I keep it at minus 20, but now when I want to use 1 ml out of it, I take it out, I thaw it, I take out 1 ml from it. I have 9 ml of liquid right now. What do I do with it? Anybody? Store it separately? Um, can you elaborate? I think you're right, but I just want to be sure if what you mean is what I'm thinking is. So when you say store it separately means 9 aliquots of 1 ml, that's a good, very good suggestion. Yes. So the thing is, I can keep that, yeah, right. So two things important. One is that I definitely need to label it that today is the day that I've opened it. If I'm using something new and I'm not finishing it, then I definitely need to write that, okay, today, 1st of September 2025 is when it was opened. And I can put it back to wherever I took it from. But now, the next time I want it, I would again have to thaw it, take 1 ml, and put it back. Now, imagine, so I do it, what, 10 times? The last time that I'm taking it, that amount has already been to 9 freeze thaw cycles. Okay. Now, there was a reason why it was recommended to keep it at minus 20 because it is stable there. Something gets spoiled when it's at a higher temperature than that. Okay. And you've given it 9 times for it to get spoiled. Every time it was out for at least 30 minutes. So what we're looking at is about what? Yeah, 4 to 5 hours is when it's been out, right? Which is not great. So what I do is what some of you are also suggesting here is once I've taken 1 ml, I have these small microcentrifuge tubes or Eppendorfs with me. I label them with, okay, that was, let's say, one of you talked about FBS. Okay, so let's say I'm labeling it as FPS. I take 9 such tubes. I label them as FPS. I put today's date on it. Okay, that or whatever the expiry is. So let's say the expiry is 4 months from now. So I put the expiry date on these tubes. You have to label it carefully. It's small space, but you have to label it. I put one ml into each, and then I put all of them back in -20. I've used one. There are 9 aliquots of 1 ml that's there. The next time, if I or anybody else in the lab needs to use it, we just take out 1 or 2 aliquots, these small um, parts of that, and use it. So the rest of it is saved from freeze thaw. Whatever we are thawing, we are using. All right. So yeah, so that's important. Same. So this goes for everything that's at -20 or -80 degrees. Okay. You would need microscopes. We talk about that briefly. Centrifuge, which you'll use to spin the sample. That would be okay. So centrifuge speed can go from 300 RPM all the way to 16,000 RPM. So revolutions per minute. Just a quick thing. The heavier the thing that you're dealing with, the smaller the rotation or revolution is what you need. When you're working with cells, 100, 200, 300 is more than enough. Cells will settle down. When you're working with DNA, now DNA is a molecule, right? DNA was a part of a cell earlier. For that, maybe 11,000, 15,000 is what you would need. This is just so that you remember, because when you're looking at these protocols, it can often become very confusing that, okay, oh, it's at 2100, it's at 2150. When it's 2150, 2100 will also work, right? It's just that it was optimized and that works. But so 200 versus 250 is a problem, because whether you're making it slower or whether you're making it faster can be a problem. But when you're talking about 11,000, whether it is 11,000 or whether it is 11,050, absolutely no problem. Okay. All right. Um, then autoclave and dry sterile. Yes. Is there a question? No? Okay. All right. So autoclave and dry sterilizer. I'm forgetting the exact specifics, but for an autoclave, you're dealing with two things: high temperature and pressure. Temperature is generally 120 degrees Celsius, and I am forgetting what pressure it is at. So if any of you remember, it's great. Otherwise, anyway, not it was not covered in this week, so it's you don't need to remember, but we can do a quick Google and see that. So so you keep your plasticware, a lot of things. Again, we'll talk about it as and when it comes. 15 lbs. Okay, thank you. Thank you so much. So, so at a particular temperature, at a particular pressure for a particular amount of time, you need to put your things so that now they are safe, sterilized to be used. And of course, then it gets all watery, it's all water around it, so moisture. So it's generally kept in a dry oven for maybe overnight for a few hours so that it gets all clean and yeah, dry, clean and dry. All right. Dry sterilizer, I have not used them myself, but from what I learned about in the recorded lectures is that it's there on the tabletop. And for smaller equipments, like your things that you are using for dissecting animals, those things you can use. So again, similar funda, but there's no water involved. So all of these things, again, would you want to have something that's your own labs, or would it be something that's there available in a centralized facility? Experiment specific instruments, again, we'll not talk about them in detail, but would there be some sort of a, I don't know, rotating thing that you need because you'll be working where you need to mix tubes and liquids, right? Would you need particular sort of centrifuges? So there are some which can work with these plates, some which can have these bigger 15 ml or 50 ml tubes versus the smaller tubes. Some that can have a temperature control on them. So would you need all of these things? Would you not need these things? Right, then we talked about whether a microscope needs to go inside that hood or can it stay outside, things like that, right? Vacuum supply, just keep this in mind. It depends. Some this may also be experiment specific, but there could be some sort of a vacuum supply that's needed. Fumigation process. I am not sure if I'm aware of. So fumigation is, another form of keeping things happy, clean, and healthy. But would you like to elaborate on that? I'm not sure if I would be able to explain this. I know fumigation takes place in labs. From what I've heard, a lot of time there are these, you know, bigger animals, for example, ants and all cases. But I don't know if there's anything in particular you're looking for. I'll just note it down. Um, so water bath in a cell culture lab. Yes, very much. So there are two options using a dry bath, which is like a bead bath. So there are these metallic beads, which yeah, okay. All right. So I'll come to both of your points. So the first thing about these water baths. So water bath is, so suppose you have, like we were talking about thawing something, right? If that's what your reference was. So certain things you never want to bring them to 37 degrees. When I'm thawing something on ice, I'm talking about bringing them from -20 to 4 degrees. If I can bring something from -20 to 37 degrees or 25 degrees or whatever the temperature of that water bath or a heated environment is, I can just put it out. So let's say generally in the labs, you have an air conditioner that's going on. So the temperature would be something around 25 degrees. So you can put your media or whatever that frozen thing is, you can keep it on your desk on the bench, and it will take, let's say, whatever, 20 minutes to thaw. So if you're taking it from -20 to 4, um, it will take a long time actually because it's still cold. If you're taking it to room temperature, if you're directly putting it on the bench, it'll be quicker, right? But then you have to be sure that you're not going to spoil that thing that you're throwing. For certain things where you're absolutely sure that it's okay, it's very safe to, thaw it at even 37 degrees. So you have these chambers what are maintaining temperatures as 37. Now, don't put things in the incubator because that is sterile. So you don't want to keep things other than cells in the incubator. But yes, another thing which holds 37 degrees temperature is some sort of a heated bath. So very much you can use that as long as you are sure what you're working with is stable at 37 for a long time. Um, so yes, water bath is one thing where you have water, and that is what your conductive media is. That is what is keeping that is what has the temperature 37. The other thing is there's also something called a dry bath or a bead bath. So where there are these beads which are warm. So instead of water, it serves a very similar purpose. You can just put your media bottle or aliquots, whatever you have in that, and so that is also what gives you 37 degrees. So you can use it. The only thing is now, whenever again we'll talk about this in more detail. So whenever you're going to actually finally use it, you need to be sure that it's clean from outside. So you just spray some ethanol on it, wipe it off with a tissue, and then use it. So that anything, because when you're working with water, let's say there was a water droplet on that tube that you have, you don't want that to contaminate what's there inside. So yeah, so water bath is perfectly good to use, but just be careful and keep changing the water. Anyway, it's going to evaporate after some time. Ah, okay. So I was not aware of how the fumigation process takes place, but yes. So let's say you have repeated contaminations in your lab. We'll talk a little bit more about contaminations, but so let's say today you're looking at your cells under the microscope, they're happy, healthy, they look good. Tomorrow you're looking at them under the microscope and you see that okay, there's something, the media is turbid, so it's a little hazy, or there's some small speck there which does not look like the cells that you are growing. So you don't want to, you cannot obtain anything from that. Don't try to salvage any part of that because even when you can't see, there would be something happening in the other part of the plate. So you have to throw it out. But throwing it out doesn't solve anything. Now you worked with it today and you saw like let's say today you saw that problem. So yesterday you were working with it. So it could be there in your lab coat now. So if tomorrow you're working with the same lab coat somewhere else, so that's a slight chance it can contaminate something. Okay. Of course, if you have kept your gloves for it, wherever you're working, clean everything up. UV, we'll talk about it, UV and whatever the disinfectants that are used, different things, there's a lot of different options that you can use. Check the incubator. This is also why you should have multiple incubators, so that if there is a contamination one, you'd want to clean it. Observe the other culture, other plates for a day or maybe, but then put them in a different incubator, clean this one clearly, put these back, so that if they were safe until now, they should continue to stay safe. So, but yes, if you're having multiple infections, right? So, for repeated contaminations, you would want to fumigate the lab. And yeah, so of course, everything needs to be super clean. So, the ethanol, everything. Uh, so, there are these, I think, formaldehyde, what you're using is, I think it's also composition. There are things like F10 available. I think that's what formaldehyde plus KMNO4 is, if I'm not wrong. But yeah, so a lot of these combination disinfectants, antiseptics are what you can use. Antiseptics the wrong word, so yeah, disinfectants, because you're cleaning a surface. And then fumigation happens, the lab stays closed for a few days just so that everything's um, cleared up. Yeah. Let me know if anything else you would like to highlight. But yeah, thanks for this discussion. This was new for me. All right. Yeah, so we talked a little bit about trash cans and waste disposal, disinfecting agents, sinks. So do not throw lab generated waste down the sinks. Um, yeah, okay. We'll talk about more in detail. What you would need, plasticware. So these things you would need in a lab, okay? This is not anything new. All right. Now, the last two things which I have with this, which we'll just spend a little bit time on. So laminar flow hood. So like I said, if I'm working here in my room versus if I'm working in an open space in the lab, that's the same thing, okay? Somebody sneezing somewhere, even if it's a meter away from me, the air is still going to be everywhere. So my first priority when I'm working with cells in a lab is to keep that cell healthy, without any contamination, right? Protected. So for that, I would want there to be no, so whatever air, so like we saw in that photo there, or what you can see here, there's this plate that you see here. Right now it's covered, but then you will open it to work on the cells here, right? Now, there would be air coming onto these from somewhere. Okay. Now that air has to be filtered. Okay. So the option is that air can come from the top, that air can come from the sides. So depending on where the airflow is from, we can call structures like these to be a laminar flow um, hood. Okay, laminar means it's a uniform flow. So that's where the word laminar flow hood comes from. Now that uniform flow, which is filtered, you see this filter here. So this is the air from the outside. This air is filtered and then it blows. Likewise, the air may be drawn from the back, filtered and then blown. So the air that is falling on your sample is going to be here, your cells here. This is the base of that laminar flow. So your sample is here. So filtered air is falling on that. Now the air needs to go somewhere. You can't just keep accumulating air. The air is going to escape. Where is it going to escape from? It's going to come out from this small part. So you see here, there's this, so backside is all metallic. And then here in the front, there is a glass slash plastic cover, which is not opened up throughout. You're opening it only such that maybe this much, I don't know how to quantify this, but maybe like 15 centimeters is how much it's opened, so that your hand can go and you'll sit there and you can work on it. Okay. You can also see it in this photo here. right? So it's open until here, and this bottom part is where it's open. So this is not a laminar flow hood. The photo, both of these are not laminar flow hoods. The photos are of biosafety cabinets. We'll come to that in a minute. But yeah, so the air is here, the air is here. Now the air is going to escape out through this opening. Now in this case, the air is already in that direction. So it's going to go out with a stronger, flow. And this is where you are sitting. So while the air which came inside is clean, but now the air has now come in contact with your cells. If you're working with cells which have no infection in them, either on purpose or by mistake, like we talk about contamination, then it's okay if this air is coming out on you. It's okay. It's not contaminated air. But what if you're working with some infectious agents, right? Or there's always this chance of contamination. Or also in general, you don't want air from these cells to be falling on you, right? So in both of these cases, that happens. So in this case, the user is not super protected. So that is where these biosafety cabinets, which we're not talking about in detail today, these come in handy. But out of these two, this one protects you absolutely zero because it's blowing air in your face or whatever is your exposed part. This one is still slightly better, but it still offers no protection to the user. Both of them are great to protect your sample, but again, you as the user are not protected. Yes. All right. So the level of biosafety, so biosafety has different levels, 1, 2, 3, 4. If I'm correct, these two fall under biosafety 1 level because they are safe, right? Um, you're still containing your sample there, but there are different levels. You can Google and see that, okay, so there are like 4 levels, 1, 2, 3, 4, one being the least safe or least. So biosafety is defined as how much safety is required for both the sample and the user. So like we talked about these infectious agents, right? Some viruses, for example, if somebody is working on the COVID-19 virus now, you cannot even work on this, right? Because while you're working on this, there's still like so much exposed area. So you can, like, there are these options, like these setups where there's just enough space for you to put your hands in. So there are these gloves which are already made. You just put in your hands there, work inside, and that's it. Right? Or also, biosafety doesn't just talk about cabinets, but it also talks about how your setup is. So basic requirement, you're wearing a lab coat, you're wearing gloves. In cell culture, it is a good practice that if your lab coat is coming till here, your gloves should be on the lab coat so that there's no exposed part of your skin that's going inside these cabinets. Because, well, your skin is going to shed cells, like dead cells. You don't want to be hovering around your dead cells on your culture plates. But again, that's still okay. You're wearing gloves, you're wearing a lab coat, and ideally glasses, goggles. But you're not wearing a mask. In some cases, and of course, it's always recommended for people who have long hair to always have them tied up. Um, but then whether or not you want to have your like a hair like something like a cap, which looks very similar to a like a net cap, would you want to wear that? What about your footwear? Of course, you're wearing shoes so that there's no spill. But your shoes will also collect all sorts of dirt from wherever you're traveling from. So would you need shoe covers? Would you need a full disposable gown? Because, well, your lab coat's just covering until your thighs, right? Your waist or your thigh. Below that is still whatever you're wearing. So would you want to cover in a full body suit? All of that comes along with biosafety levels. So biosafety 1, most common. Biosafety 2, also quite common. 3 becomes rare. 4, super rare. There would probably be like a handful of institutes, handful of laboratories around the world which can handle this sort of a level, right? So of course you need to get trained in that. Um, and you have to be super careful. In a lot of these places, you need to take a shower after you've done your work and things like that. So it's good. You just it's a good thing to look at. If I get some, I remember watching a video sometime back which explained a biosafety level 4 setup. I'll see if I can find that and I'll send you a link, or maybe I'll add it to these lecture slides. But yeah, I think if you Google it, you'll be able to find that. So yeah, so horizontal versus vertical laminar flow. Vertical is slightly better, but it's still, both of them still don't offer any protection to the user. Typically, what you have in them is this. So blower, of course, this is where the direction of air is being controlled through. Light, here we're talking about just light, because as you can see here, there's this tube light inside it, because you don't want to work in the dark. You can. Because I remember having this discussion with somebody where they said that when you're working with cells in your body, especially when you're talking about non-skin cells, the other cells have no idea about what light is. Inside your body, there's no light. So an ideal working environment for cells is without light, right? And that's how they are in the incubator also. There's no light inside the incubator. So why should you have tube light here? But well, you need to have light so that you're able to work in these systems. So there's a tube light. It's up to you if you want to switch them on or not. And of course, there's going to be UV, which is ultraviolet light. Now, ultraviolet is super dangerous. So you should not be anywhere close to that system where the ultraviolet light is on, nor should your cells be there. But before and after, or before or after, depends. This can be very lab specific, work specific. You have to switch on the UV light for about 30 minutes or so. So in a lot of places, what is the general practice is once you're done, you UV the cabinet, you close it and you UV the cabinet for some time. So it's ready to use for the next user. In some cases, if you're not sure if the person before you has used it, then you will do it before 30 minutes before you have to start. A lot of these newer cabinets, what they do is as soon as you pull this glass shutter down, it automatically switches on the UV for a set amount of time. You see all of these, there's some buttons here. So you can just automate that and say that, okay, every time I'm closing these doors, it should UV for 20 minutes. Yes, good question. Just give me a second, I'll have a sip. So again, it could be a matter of preference. Now, the things, while they will be safe when they're inside, so your cells and your media, ideally all of this should be out. Your plasticware, maybe some of your glassware, these can be inside. Now, why should they be inside and why should they be outside? So, first of all, if they're outside, you're not sure if they're sterile or not anymore. Okay. In cell culture, again, it depends on the extent of contamination you get in the lab or not and how careful you want to be. So I don't know if you can see here, these are filter tips. So these tips have a filter on them so that nothing falls onto these tips, so that nothing goes into your media when you're using these tips. Now taking them out, putting them in while they're closed, it's all good. But again, that adds up to how many times you're taking it out, bringing them back in. When you're UVing it, oh, sorry, before I go that, so if these are kept inside at all times, and if I have used, then when the next person comes, they're also going to use the same thing. And the cycle goes on. If anybody messes up, everybody else's things can also get messed up. So either you can label yours, but then what happens is I would need 4 different sizes of tips. Person number 2 will also need 4 different sizes of tips. I'll keep on adding up the capacity of this hood or this cabinet, right? So cabinets are small, okay? They can be small, maybe like up to, I think, yeah, the same. They can also be like up to 3 feet wide, 2 feet wide. They're not super big, right? So you don't have unlimited space there. And as you can see, this looks already very clustered because you'll have 3 or 4 different pipettes, tips, you'll have these ependoffs here, you'll have your media, you'll have your plates, you might need a few more things here and there. So it takes up space. So whether or not you take it out, it depends. from what I have seen in my work, we keep it inside. Everybody uses the same thing. Once something is finished, you replenish it. You take one newer one from the shelf, from the cabinet, from the storage cabinet. So, yeah. And yes, every time something is brought inside the cabinet, inside the hood, it needs to be sprayed with ethanol and wiped so that you're not bringing along any dust, anything with it. And when you spray ethanol, there's a good chance that everything that you've marked on this, that's going to go off. I mean, I don't know if you can see, but then here there's a little smudge, right? The whatever it was labeled as, it's got smudged. So people use ethanol resistant markers for that. Or if it's not available, then every time that you're spraying it and every time the label is going off, you pre-label it. That's okay. You get with it, you get accustomed to it with time. So, but yes, so these things inside the cabinet, what you would normally definitely need, whether or not you're keeping it inside every time, or whether one set is there always inside. These would be pipettes and pipette tips. There would be a holder, generally like a bigger one to keep these tubes, and a smaller one to keep your microcentrifuge tubes. Some people like to keep like a scissor inside. Sometimes you would want to cut the base of your tips to make it a wide bore tip. Again, so things like that. You may want to keep a marker or two inside. These Eppendorfs can be inside. So yeah, so those things are generally what are there inside the cabinet. So every time that you're using these cabinets, again, UV and ethanol. These two things, it depends. They need to be done at least once. If you're not doing it after you do it before you're starting work, or if you're not doing it before, then ensure that everybody does it after they are done so that the next person can just directly start using it. But again, it's your preference. Another thing which is not visible here is, so when you're done with this media, so here, if you can see here, the media looks a lot yellow, right? So, and I don't know if you can see here, this is also media. This looks quite pink. This is fresh media and this is used up media. So this has been probably 2 days that the cells have used up the media and it's all now acidic. So I need to remove this media. Where do I remove it? So I can use a pipette, take it out and put it maybe in one of these falcons and then trash it. Or I can have a bigger bottle where I keep stocking up that trash and then throwing that once it's full. Another thing, I talked about using a vacuum here. So a lot of these cabinets, if you see here, they have a vacuum setup. So what you can do is, on one end of your vacuum is you have a tip, and on the other end of that vacuum, like another pipe goes on into one of these storage containers. So you can just suck, so that just sucks up this media, right? So you don't have to take it in, throw it, take it in and throw, but you can just keep sucking up the media. Again, all of these are different ways to do the same thing. Whatever's available, whatever works out, resources wise. Okay, this could be available in different places. But yeah, so that's what are the things inside the cabinet. And we briefly touched upon biosafety levels and cabinets. So yes, so again, laminar hood, vertical and horizontal. Vertical is slightly better. Okay. The final thing that I think what we'll be talking about today, penultimate, just second last thing, liquid nitrogen facility. So this is the sort of big container, the tanks, to keep your liquid nitrogen and your samples in. Again, there are multiple different options. If there's a centralized one, then this would be this big. If there are smaller ones, then they could be small like this. Either way, there would be slots to keep boxes of cells inside it. Boxes, we'll just call everything cryo. So the prefix would be cryo. So cryo boxes, cryo tubes. You have to, so these boxes, they don't get damaged, or they're not cardboard boxes. So they're not going to get, wet every time you're taking them out, putting them back in. Tubes also, they're slightly different than your normal Eppendorfs. because they need to withstand a lot higher, like a lot lower temperatures. And you don't want them to blast open as soon as there's a difference in pressure, which happens. So there are these rotating cap ones rather than the clip type. Um, and you generally have a larger space to label these tubes. So these are not pointy at the bottom, but they're more cylindrical. So there's a good space for you to label. Now, labeling anything you put in liquid nitrogen. Okay, so I'll come to it in a second. So you're talking about very, very low temperatures. Liquid nitrogen, there are two ways in which your cells can be kept there. One is you're submerging the cells in these boxes in liquid nitrogen. And the other thing is there's liquid nitrogen at the bottom of these tanks, but then everything else is just vapor liquid nitrogen and it's still cold. So either way, that's liquid nitrogen. So cells are kept in that. So remember we talked about -20 being a good temperature for something, -80, it's better for RNA. This is -192 or 196. Oh, I think it's 192. So that's, anyways, that's for yeah, for your cells to stay. So let's say today I'm working with some cell and then I'm done working with them, but I spent a lot of time making these cells what they are. I might have to use them 6 months later when, I'm publishing a paper and the reviewers say, okay, do one more experiment. And I'm like, oh, I don't have the cells anymore. So what do? I go to this liquid nitrogen facility, I pull out my cells, I start growing them again from then and there. I don't need to do all the extra work again. Okay, I can make multiple of these stocks. These are called liquid nitrogen stocks. Maybe I can make 10 of them, and then I can start my experiment 10 times if need be. So it arrests metabolic activity and cells also. So cells are not dead. They are just in some sort of a sleeping zone, sleeping phase. But you don't want the cells also to burst open, just like these tubes. So DMSO glycerol, there are these again, either directly DMSO glycerol or there are also pre-made cryo mediums where you can take, let's say 10,000 cells, put them in half an ml of that medium and put that in these vials and then put them in -196. But there is a catch here. If you put them directly in 196 or even -80, that may not be great. So what you do instead is you have to bring them in a controlled manner. You want to cool them in a controlled manner, which is roughly like, so if I keep myself directly in the fridge or in the freezer, they're gonna come to -80 super soon. From 37 degrees to -80, if it takes them about 2 hours, then that's the perfect time. So there are these small boxes which are generally filled with isopropanol, if I'm not wrong, and styrofoam or plastic and different setups. So you can put these cryo vials in those boxes, not this box, but in those specialized boxes, and put that box in -80. So what it does is it's going to take the temperature slowly to -80. And then for -80, you can put them in liquid nitrogen directly because now they're already super cool. So it's okay. So from 37, you put them in these tubes. These tubes go in those special boxes. And maybe the next day or after a couple of hours, you can put them in these cryo boxes. And this box goes into one of these slots. So there are multiple slots, maybe like 6 columns of, let's say, 12 boxes each. And each box here can hold 100 tubes, so 10 by 10. And then now you've made 10 vial stocks today, you make 50 tomorrow, and so on. It's going to be very, very hard to remember 6 months from now what you did. So organizing this, so don't arrange it randomly, arrange it in a row and keep a note of that. So of course, these need to be labeled well, but so should you have them labeled with you. That okay, on this date, I put 10 stocks of this. They were in this media. I took 10,000 cells. I put half an ml of whatever the cryopreservation media was. these were growing for 4 days. This was we'll talk about it, passage number, blah, blah, blah. And then if tomorrow you take out one of these vials, you make a note in that same table that, okay, I had 10, now I'm remaining with 9 of them. So that if you come down to 4 or 5, you know that I need to make more stocks. You don't never want to come down to 2 or 3 stocks. It's very risky. Okay, so am I missing something? Yes. So now liquid nitrogen will also evaporate after some time. So this needs to be refilled. Again, whether it's a centralized facility or whether it's in your lab, if it's you who needs to do this, then you need to bring liquid nitrogen from a centralized facility and put that in your tanks and do that. Oh, hemocytometer. I will come to it in 1 minute. Okay, good question. So yeah. Okay, final thing. Let's say you have this stock with you, right? You made this last year and now you want to use it. So these things you have them in liquid nitrogen. Let's say you have liquid nitrogen in your lab. You have that. Quickly you want to thaw them at 37 degrees. Quickly, you don't want them to come to 37 degrees because, oh, would anybody like to try? So I said that when you're bringing them from 37 to minus 80 or minus 196, you're bringing them 1 minute, 1 degree at a minute. That's how you're cooling it. But then when you want to thaw it, you're thawing it rapidly from minus 196 to directly 37 degrees in that water bath. Why? Would anybody like to would like to try this? Any guesses? No? Okay. To prevent ice crystal formation, that's one thing. Yes, true, true. And another thing also is that now the cells are waking up. Okay. They're now getting more metabolically active. And now when they're getting at a temperature closer to 37, what they are seeing around them is not food, but they're seeing DMSO. And DMSO is not the best for cells. Okay. DMSO was good to keep them preserved, but now you don't want that. So what you want to do is you want to quickly thaw them and then quickly put them, like centrifuge them. Generally, that's what's done. You thaw them, mix them with a little bit of the media, centrifuge them, remove whatever that supernatant is because that had DMSO in it, throw it. Now mix them in absolutely fresh media and then put it in your plates. So that now, very, very soon, they get their food and their nutrients. All right. So that's that. All right. Okay. So yeah, so I'll just to make a note, I'll just put this here that the fine needs to be quick at 37. All right. Okay, so we have covered that. Let me know if there's any question. And then hopefully the final thing for today is whether or not you're using animals in your work. Again, I have not worked with animals, I've only worked with cells. So I won't be explaining any extra here. So things that need to be considered is how many animals would you need? So oftentimes experiments need to be run in replicates, right? You cannot just show that, okay, one animal shows a difference, so that's it. You need 2 or 3. How will they be housed? Again, is it a centralized facility? Is it in your own lab? Who will take care of it? What are their food requirements? What are the water requirements? Airing, cleanliness, right? So how do you manage that? Are you trained for it? Would there be somebody else taking care of it, care of this whole system? How are they sacrificed? So in the lab lingo, the word used is sacrificed. So they can be like that can be through CO2 inhalation. That can be through anesthesia overdose. Once that happens, they can be decapitated. So, like the head is removed or whatever is removed. Cervical dislocation is the cervical bone at the back, the spine that's dislocated. So, it all has to be very carefully planned out and cleared by the ethical committee. Every institute, every department has an ethical committee to whom you have to present, and that is the sort of thing you have to do. There is a term that I would just like to introduce to you if you've not already heard this. It's there are like these three R's. You call up you. It's like in the other regime, it's called reduce, reuse, recycle. In case of animal use, it's called replacement. So, for example, do you have to use an animal, or can the experiment still be done on cells? Reduction, do you need six animals, or can it be done in three? For example, do you if you're working with something like sex differences, so does it behave a certain thing, does it behave differently for males and females? You of course have double the sample now. But if you're not interested in, or if you're sure that whatever your setup is, it's not going to show sexual differences, then you don't need that double number. So it can just be, so a reduction in that way. And refinement is how can you make things better for the animal? How can it be less painful and all of that? So that's the three R's in animal use. And with that, we come to an end of this week's discussion. This was a long one. I think we've already been it's been to 1 hour 45 minutes. So we'll quickly, I think I just have 5, 6 questions, but most of these we already covered in this. So most cell lines, plural here, most cell lines, they grow well at what pH? Quickly, just put 1, 2, 3, 4 or A, B, C, D. So yeah, 3. All right, perfect. Next up, in culture media, what is used as a source of what for glycolysis? So glycolysis, the name suggests, yeah. So using up of glucose, using up, it's yeah, using up glucose. Yes. So carbon is used as the I think it should be, yeah. It's the other way actually. So it's glucose is used as a carbon source, right? It's not carbon as glucose, but yeah, glucose as carbon. Good. So confusing question, but just read all the answers, all the options carefully. Right. Which PPE, which is personal protective equipment, what we talk about mostly, what is essential when you're handling mammalian cell cultures? Yes, so it's one. We talked about it, so you don't necessarily need a face mask because you're not working with infectious agents. Full body suit also not required. Shoe covers only is not. Shoe covers is an added thing. So it's again it's a it depends how important that is. But yes, lab coat, gloves, and safety goggles are super important. Higher level of oxygen in cell cultures could be toxic due to. This is more relevant to what we talked about last week. But why is a higher level of oxygen a problem? Because it increases free radicals. Good. Which of the following is used for the sterilization of wood? So there are two answers which are correct here. UV and alcohol both. Okay, so alcohol, ethanol, yes. So I just replaced one of the answers, one of the options, just so that you have two options correct. And both of them are actually important. All right. So great. And then we talked about this 4 feet by 2 feet roughly. Why is that a recommendation? Working space. And here we are not talking just about incubator or like not just about the hood or anything, but yeah, because you want enough space because you don't want any accidents happening. You don't want people bumping into each other and their plates falling. All right. Oh, one more question. So yeah, which of the laminar hoods does not throw air at the person who's performing the experiments? Yeah. So for the purpose of today's discussion, we will say vertical laminar flow. It's not throwing air at the person, but it's still not perfect because the air is still going to the person without being filtered. But yes, it does not throw air. So vertical is good. But if the question would have been which of these laminar hoods protects the person performing experiment, then it would be none of the above because you're not being protected as such, right? But yes, vertical, that's the correct answer. Don't get confused. And that's about it. So we can talk about any questions, any doubts that you may have from assignment one. So the solutions are released for every assignment, I think on Thursday or Friday. You get a PDF document which discusses all the answers and why that's an answer. So if there's any, so take a look at those solutions. If you want me to discuss that, let me know because, but I would just say that it's been almost 2 hours already. I don't want to take up your time unnecessarily. If you say you want me to discuss, so have it open. Otherwise, take a look at it. We can also talk about it next week, or you can just put any questions you have from that assignment into that Q&A thing. Uh, oh, so live cells in tissues. Okay. So yes, so the thing here is while you're using a microscope, but you're not, what you're doing still is the live cell is in tissue. Had this been live cell in a cell culture dish, then that's a different thing. But when you're still talking about that tissue, so tissue is still your um yeah, it's still the real thing, and you've not fixed them. They're still very much alive in that tissue. So, okay, let me open the assignment solution then. For those of you who would like to not attend, if you want to leave, you're free to leave the session. So I just want to tell that the assignment for this week's assignment is day after tomorrow, and this week's session, like another session next week, would be on Monday, 8th of September. We have recorded the lecture. It's going to be uploaded on YouTube, and this PPT will be added to the Google Drive. And let me just open this thing then. So yeah. So what you had talked about here, yeah. So this question, right? Using a microscope to see real-time imaging of cells and tissues, it says that live cell imaging is done without taking the tissue out of the system. It's in vivo. So it is a confusing question, but I think the main crux of it was live cells and in tissues. So that's what classifies as in vivo. But yeah, I can understand why it's a tough question. And then question 13, which was this antioxidants one. So again, the thing was that antioxidants are what reduce the oxidative stress and they eliminate free radicals. So one of these is anthocyanins, which also is a acid-base indicator. So it's red and acidic, blue and basic, based on the surrounding pH. So what the researcher is doing is that they're developing some colonies in the presence of an oxygen-rich environment, and these colonies, in the presence of this oxygen-rich environment, they release anthocyanins to eliminate the free radical. Now, if you are measuring the optical density of these anthocyanins, that is, optical density is proportional to the intensity, to ensure the system is working, what would the researcher observe? So the answer that's correct here is initially the OD will, so when it's introduced into the system, right now it's just going to, anthocyanins are being released there in like initially. So the anthocyanins initially have a that optical density is increasing, but then immediately they are going to take up all of that. They're going to reduce the free radicals, so that's going to decrease. So it increases initially, but then keep on decreasing. So that's again, these two are some of the confusing questions here, but you have the explanation here. if this is still not satisfactory, I would, if you have another explanation for what you think is the right answer, I think the best way to go for this would be putting it on this Q&A forum here, because I have not set up these questions, and it is possible that the questions can have more than one correct answer depending on what sort of a assumption you're taking. So if that is the case, I would recommend you to put this here. But if you're satisfied with the answer, then this is what there is. Any other question? If not, then we'll finish the session here. Thank you everybody for joining in, and this was a very interactive session. Great questions asked. I will check the copper sulfate in water thing and let you know next class. But until then, thank you so much, and I'll see you again next week. So yeah, bye-bye. All right. Stopping the recording and everything. Yeah, bye-bye.